All right, in this lesson we are going to look at mutations. So the first thing you need to look at is that mutation, by definition, happens when a sequence in DNA is changed. And we've talked about this when we talked about cancer and we looked at what causes mutations. There are two types we're going to look at, and I know your note sheet says 3. We're going to not worry about the inversion. The inversion is a different kind of mutation. We're not going to worry about it. We're only going to look at 2. The first is a point mutation, and the point mutation is also called a substitution. Okay, so a substitution mutation. In other words, if, for example, you had a sequence that read ATG, CCA, and we changed that to read A, T, G, C, G, A, then you would have a base that is different from the original, right? We've substituted in a different base at one point. That's the point mutation. Now, here's the thing. These are generally going to only impact one amino acid, right? So we generally look at these as the least of the harmfulness in mutations. So only one of the amino acids, the one that ultimately gets coded for by that 3-nucleotide sequence, is the one that's going to be probably a different amino acid. However, let's look at a situation where a point mutation does something different. Let's say our DNA reads G, A, G, A, A. Okay, so our original DNA sequence reads G, A, A. And we mutate it so that now it reads G, A, G. Okay, so we've now changed the last letter. That's a point mutation. Now that's our DNA. If we go and we look at the mRNA sequence for this, right, that becomes CUU, right? This one becomes CUC. Okay, now let's look at what that ultimately is going to do to our amino acid sequence. If we're reading the chart, remember, CUU, this is what we're reading, that was the original sequence. CUU is going to give you leucine, right? Remember that from yesterday. So this is going to be LEU, whoops, leucine. Okay. If you look, though, and you find what CUC is instead, CUC is still going to be in the same spot there. It's still going to be leucine. This is what we call a neutral mutation or a hidden mutation. In other words, you've changed from the original sequence. Okay, the original sequence is going to ultimately impact the messenger RNA molecule, which could potentially impact the amino acid that you put into that spot in the molecule. However, sometimes, because there's all this code overlap in our chart, sometimes you can make a change and you still end up with the same amino acid. So this is a hidden or a silent mutation, and it turns out this ends up not being at all dangerous because it doesn't change the amino acid, it doesn't change the protein. Only if the mutation ultimately changes the protein is it a problem. All right, now the next one we're going to look at is called the Frameshift mutation. Okay, so we're going to look at the Frameshift mutation next. And you'll see why it's called a Frameshift mutation. There are two types. There's the deletion. Oh, and I messed that up. It's not substitution. Let me cross that out. It's either the deletion or insertion. Okay, so either the deletion or the insertion kind of frameshift mutation. All right, so let's look at both. Here's our original. Ready? This is what the original piece of DNA read. ATG C, C, A. All right, if we did a deletion, so if we delete one letter, this becomes A, T, and say I cut this G out right here. Notice all the letters shift down to fill in the codon. So this C that was here ends up shifting over to replace the missing G, okay, which is what you're seeing here. And then you're left at the end with this incomplete codon that doesn't have enough stuff in it. Okay, that's why it's called a frame shift, because all the letters shift down. Here's what an insertion looks like. Okay, in this case, instead of deleting a letter, let's put in an extra T here. Okay, again, what happens is there was no T in our original sequence. Okay, so up here you didn't have a T, so the extra T that we put in that goes right here is going to force all these other letters to shift the other way. So here's that. Okay, so again, you're either way, whether you insert or whether you delete, you're going to cause a shift. The problem with these shifts is from the point where the mutation occurs, every single codon the rest of the way down is going to be messed up, which means every single amino acid the rest of the way down is going to be affected, and usually negatively affected. So the problem here is that frameshift mutations tend to be really, 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 really bad because your protein doesn't resemble anything like it used to. At least with the point mutation, you only mess up one amino acid. If you're talking about a frameshift mutation, it gets really, really bad really, really fast. All right, so let's summarize what we know now about mutations by saying, in general, the point mutations are the least harmful, followed by the frameshift. So these are more harmful. And it doesn't matter which frameshift you're talking about, either frameshift is going to be harmful. There is one little statement, though, that we have to add to that, because any mutation in one of two codons that are special is going to be harmful. And the two special codons are the start and the stop codon. So any mutation in a start or a stop codon is going to be harmful, because if you mess up a start codon, you don't get to start. If you mess up the stop codon, you're going to get a much longer protein than you anticipated. And that's mutation. There's really not that much to it. Let me give you just one more thing before I leave you, and this is also on your note sheet. There's nothing really to write here, except you'll probably want to copy this in your notes. The old biology book, the old version, had this example, and I really like it because you start with a sentence made entirely of three-letter words, right? And it kind of just shows you how changing that sentence, changing one letter versus dropping one or inserting one makes a difference. So here's our original sequence, right? The original sentence reads, "The dog bit the cat." It makes sense. Everybody gets that image in their head. You can make a point mutation in it by changing just one letter. So now instead of "the dog bit the cat," the sentence becomes "the dog bit the car." Notice you've only messed up one of our words, and it's not terrible. Okay, it's not awful. Now look what happens if we drop the E from "the." Okay, remember all the other letters are shifting in this direction to try to fill in, and notice that you don't even have anything that looks like words anymore. Same thing here. At this point, if we insert a letter, an extra E in, it shifts everything in this direction. And again, notice from the point of the mutation, in this one from here onward, in this one from here onward, every other word doesn't make sense anymore. Okay, so you can kind of see from that exactly how harmful these mutations are.